Hi there. In this video, we'll continue our discussion of protein transport, describing ways that proteins get places other than the nucleus or mitochondria. The fundamental mechanism for getting to other organelles is by a vesicle budding. We'll show that the launch point for all these destinations are having proteins synthesized directly into the ER. From the ER, the vesicles will bud through the Golgi stack, which is a quality control organelle and a place for sorting new locations. We'll talk about protein secretion out into the extracellular space. We'll also talk about endocytosis, in which materials are taken up from the outside, and we'll conclude with a little bit about the function of the lysis, a key organelle for the recovery of biomaterials. So this slide emphasizes the fact that proteins that will be in the endoplasmic reticulum will transfer by vesicles to the Golgi apparatus, and then they'll be sorted. They can be sorted for extracellular space, they can be sorted for the membrane itself, they can be sorted to combine with endosomes taken up from the outside to lysosomes where materials are recycled. More than a third of human proteins are either plasma membrane proteins or proteins secreted to the extracellular space. So this is often used mechanism by the cell. So to get to other locations, we have to first get the protein into the endoplasmic reticulum or the ER. And as you can see, this will look a little bit like the mitochondrial case where we're threading a unfolded protein through a pore into an extracellular space or the membrane. There's a big difference though between this and the mitochondria. In the mitochondria, to get proteins in the mitochondria, we synthesized a full-length protein, unfolded it, and then pulled it through the pore. The chaperone proteins did. In this case, only a very small fragment of a peptide chain will grow. The signal sequence that indicates that this protein is destined for the endoplasmic reticulum will be bound by a protein, and this will pause translation. It's going to make this peptide and it's going to stop. This protein will interact with a receptor in the ER membrane, which will end up positioning the ribosome over this translocation channel, and then protein synthesis will resume. So you're actively synthesizing the protein into the endoplasmic reticulum. So now we have to talk in a little bit more detail about the signal sequence. If a protein has this signal sequence and no others, and we'll see them on future slides, that means the protein is going to get translocated all the way into the lumen of the ER. The signal peptide will be cleaved, and this is a soluble protein, and one very likely destination of this protein is secretion to the extracellular matrix. In almost all cases, the signal sequence is cleaved from the protein by a peptidase. Then the protein will be released, the signal peptide will be released, later on degraded, and the translocation channel will close again. On this next slide, we start to see a slightly more complicated set of signal sequences. Here we have the two signal sequences, the leader sequence, and also an internal hydrophobic sequence that's called a stop transfer sequence. And this leader sequence will function as before. It will open the translocation channel. It will signal to the ribosome to continue to synthesize. The ribosome is not shown here for clarity, but it's sitting on top. It's synthesizing the protein into the ER. And then when this stop transfer sequence hits the translocation channel, this will be the signal to stop translocating. The ribosome will finish synthesizing the rest of the protein, and the leader sequence will be cleaved off. But now you have a protein bound in the endoplasmic reticulum membrane. One likely destination for this protein is a protein that is stuck in the outermost plasma membrane. In this case, the carboxyl group is on the cytosolic side, right? This is cytosolic, and the amino side of the peptide chain is in the ER lumen. When the vesicles fuse and bud, the orientations are maintained. The cytosolic side always points to the cytosol. This amino end, which is now in the ER lumen, will be the part of the protein that's exposed to the outside world. A very large number of proteins have a single alpha helix crossing the membrane with an exoplasmic domain and a cytoplasmic domain. But we'll see more complicated geometries as well. On this slide, we see an example of a more complicated geometry. The final product will be two transmembrane helices, a loop that will be sticking outside in the exoplasmic side, and both the carboxy and amino sides will be sticking on the cytosol side when the protein reaches its final destination. In this case, the start transfer sequence is internal, so that meant the ribosome was making this peptide, and it didn't pause until it got to the start transfer sequence, which was bound by the signal recognition particle. And then it starts to act just like the leader sequence does, but since this is internal to the protein, it won't be cleaved. This will continue until the hydrophobic stop transfer sequence arrives, and that will be the clue to the protein import machinery to stop translocating the protein and to keep this helix in the membrane. Again, this information transfer or signaling of this is where it should be contained in the membrane is pretty simple thermodynamics. If you have a string of hydrophobic amino acids in the middle of a protein, that is going to be a signal to, in this instance, to stop the transfer. While this signal is fairly specific, there are a wide variety of possible amino acid sequences that will stick in the endoplasmic reticulum membrane. Now, the proteins move between these different compartments via vesicles, and the vesicles are coated by certain proteins which help direct traffic. There are three proteins we need to know. We'll start by talking about COP2. This is the protein that will coat a vesicle to direct transfer away from the nucleus. The nucleus is over here. Here's the plasma membrane, so vesicles coated with COP2 go away from the nucleus. Vesicles coated with COP1 go towards the nucleus. And then vesicles coated with clathrin are specifically involved in endocytosis. They will fuse with endosomes and possibly lysosomes, but they can also end up as part of the Golgi network. So how do these COP2, COP1, and clathrin proteins help the vesicles movement? Well, they interact with different types of motor proteins. We'll see in a future lecture about how microtubules are basically the highways of the cell. Microtubules will generally extend radially from near the nucleus outwards in a starburst pattern inside a cell, and different types of motor proteins travel different directions on microtubules. So in the next slide, we'll discuss how vesicles, once they've budded, know how to fuse with a destination membrane. The proteins responsible for the fusion, directing the fusion of the vesicles, are proteins called snare proteins. There's a V snare protein, which is a vesicle snare protein, and a T snare protein, which is a target snare protein. And these are proteins which extend out long ways, and when they come in contact on the tips, then they start to zip up, and as they zip up, then this is bringing the vesicle closer and closer in apposition to the membrane. And once you make contact here, you'll have spontaneous thermodynamic fusion. This high degree of curvature is unfavorable, so this will flatten out, and eventually you'll have integration of all the lipids in the vesicle to be contiguous with the target membrane. So again, this process is driven entirely by thermodynamics, the binding energy between the V snare and the T snare, which as they encounter increases as these proteins zipper up, and then the fusion of the membranes occurs. And specificity occurs because there are a wide variety of snare proteins, T snare proteins. Well, there's a compartment A and a compartment B, and you see there are different snares. This one has sort of a round shape. This one has sort of an angular shape. Different V snares are going to indicate that the destination for these vesicles are, in the round case, compartment B, and the triangular case, compartment A. There are about 60 different snare proteins in humans, And so that's the number of locations that can occur. And they were in fact the subject of the most recent Nobel Prize in Medicine. A few words about the Golgi. Many of these details we do not need to know for the test. Is that this is where protein sorting goes on. And one of the things that in this stack occurs is the addition and removal of lots of different sugars, since most of these proteins are being secreted or sorted to plasma membranes, and most of those proteins are glycosylated, as we said. The cis face faces the nucleus and the endoplasmic reticulum. The trans face is towards the outside world, the plasma membrane. And another function of the Golgi is quality control of protein folding, but that's a bit of a detail. Okay, I think this will be a good place to pause, and we'll resume in the next video. Thanks.